Okay, good morning. My name is Marty Burke. I am at the University of Illinois at Urbana-Champaign.
And my name is Matt Long. I'm at The Ohio State University in Columbus, Ohio.
We want to welcome everyone to the symposium this afternoon, the workshop. Thank you so much for coming. We all understand that, of course, the modulators taught us something incredibly important, that CF can be treated in very effective ways in restoring tremendous amounts of quality and extension of lifespan to those who need those medicines. And yet, we're all acutely aware that there are many people who still cannot benefit from modulators. And so the inspiration and the motivation for today's workshop is to take some time together and think very carefully about other ways in which we may be able to restore function and thereby hopefully provide the same benefit to those who still can't benefit from modulator therapy. One of the things that we thought a lot about when we put the session together is the importance of kind of multi-strategic thinking and for trying as a community to solve a really important, very challenging problem. It's critical that we think differently and that we embrace and celebrate people who are courageous enough to think differently. And so we were very excited to select talks today of individuals who are doing exactly that and showing a range of different strategies, different ways to try to approach this really important problem. And we think that is critical for ultimately as a community together finding a way to make a difference. So what you're going to see is some fantastic talks from researchers who are taking different approaches, okay, to try to get back meaningful benefit and function and thereby provide benefit for those who can't benefit from the modulators. We're going to think about different strategies that span the gamut. So perturbing, other ion channels, thinking about how we might be able to restore CFTR through mRNA, thinking about ways in which we could actually do a molecular prosthetics approach to provide back that missing activity, and also, of course, the very important strategy of looking at read-through compounds. Okay, so you're going to see a whole menu of different approaches. We're hopefully it's going to stimulate a great discussion. We're looking forward to the question and answer sessions, and use this as a forum. Okay, I think there's been time and time again, one of the many things I love about NACFC, there's so many times where someone asks a great question, right, probes an area, maybe say, have you thought about it this way or that way? And then it leads to really a spark and a new idea that then we hear about next year as a really exciting, different approach or something that's making progress. So you play a really important role here today, and we're looking forward to this being a dialogue and a great discussion and hopefully stimulating a lot of creative ideas. Okay, I'll turn it over to Matt and we'll get us started.
Thank you. So today we have 5 different talks. They're going to be 15 minutes in length, and then we have, I believe, 8 minutes for Q&A. So if you're in the room and you have a question, please feel free to queue up at the microphones after the talk. And please just state your name and where you're from so the speaker can know who you're talking to. There is also a chat function in the app that we'll try to monitor for questions. So, and yeah, if you can't hear the speakers, please let us know and we'll try to turn up the microphones. So the first talk, I'm happy to welcome up to the podium Emily Bulik-Sullivan, who's a graduate student at UNC, and is going to be talking to us about elucidating the mechanism of ENaC activity reduction by CC90009. Thanks, Emily.
I think I'm a little shorter than our moderators, so hopefully you can hear me. Is it working? Okay. So good morning. Thank you. I'm Emily Bulik-Sullivan. I'm in the MD PhD program at UNC Chapel Hill, and I'm going to be telling you about the work that we're doing in Scott Randell's lab, trying to figure out the mechanism by which this small molecule, CC90009, I will call it CC9 because that's a lot of zeros, reduces ENaC activity in respiratory epithelial cells. I have no disclosures. So it almost feels unnecessary to give this background to this particular group, but as all of you probably know already, there are tons of documented CFTR variants, only some of which are disease-causing, and they're grouped into 6 classes, but not every variant is eligible for modulator therapies. So there are some people with CF who, because of their variants that they harbor, cannot take and benefit from modulators, or there could be issues with availability of these drugs or the cost or whether they can tolerate them that render them ineligible to benefit from modulators. So there's this push and this interest in finding variant agnostic therapies that would allow more people to benefit regardless of their CFTR variants. And so I'm the perturbing the ion channels person that they talked about in the intro. So we are looking at the epithelial sodium channel as an alternative protein that we can work with trying to find therapies for. So this is a massively oversimplified diagram of the respiratory epithelium. This is like an example of one cell. And you can I point? Oh, yes. You can see here that CFTR in this diagram of a non-CF airway is active in allowing chloride and other anions to exit the cell. ENaC is active in allowing sodium to enter the cell, and in tandem, they work together to maintain the hydration status of the mucus atop the respiratory epithelium. And then, in converse, in a CF airway, you have a less active or completely inactive CFTR, and ENaC is thought to be hyperactive, and then water follows salt is pulled into the cell and out of the mucus. The mucus becomes thick and sticky, and that causes the hallmark pathologies of CF. So we are interested in trying to figure out drugs that could be used to reduce ENaC activity as an alternative because this is such a complicated thing to find agnostic therapies for. The drug that we work with, CC90009, is a derivative of this compound here called thalidomide, which I think many people are familiar with, but it's sort of a classic cautionary tale in drug development history, so it's worth going over the past of this compound. So in the 50s in Europe, it was prescribed for pregnant people who had morning sickness, but was rapidly discovered to be teratogenic because they would give birth to babies who had phocomelia or limb abnormalities. So it was removed from the market. This woman here is Frances Oldham Kelsey, who worked at the FDA at the time and did not approve it for use in the United States, and she was very highly rewarded for her wisdom in that matter. But then in the 90s, it was reintroduced as a repurposed drug to treat mostly hematologic cancers, obviously with a black box teratogenicity warning. And then in 2010, a lab in Japan identified that thalidomide binds to cereblon, which is a component of an E3 ubiquitin ligase, and that's both the mechanism of action of the drug and also the cause for its teratogenicity. So since all of this has gone down, clever chemists have modified the base structure of thalidomide to try to repurpose it and expand its use into different conditions. Thalidomide is a heterobifunctional molecule, which means it has two parts. This glutaramide structure here binds to cereblon and anchors it to the E3 ubiquitin ligase. And then the thal oil group binds to whichever neosubstrate you're trying to link to the E3 ubiquitin ligase and cause to be ubiquitinated and then degraded. And they decorate the thal oil component with different functional groups, and that tweaks what neosubstrates the drug will bring to the E3 ubiquitin ligase and cause to be degraded. And so we work with this drug CC9, which has this long functional structure here, and it has a known mechanism of action. So it was first identified and published about in 2021. And in these papers, they were using it to try to treat acute myeloid leukemia. And they also identified a known mechanism of action. So this is the Col4-DDB1 cereblon RBX1 E3 ubiquitin ligase. There are hundreds of E3 ubiquitin ligases, and they're all specific to particular proteins that the cell is trying to recycle. So this is just the one that this particular molecule works with. And so this binds to cereblon and causes this compound ERF3A, or the eukaryotic ribosomal release factor 3A, to be ubiquitinated and then proteasomally degraded. And I am not a cancer biologist, but in cancer, that's valuable. Don't ask me questions about leukemia, please. And so CC9 was in clinical trials, just phase 1 for a while. They have since ended. This one because business objectives changed, and this one because it wasn't efficacious in the AML patients that they were trying to treat. But it's encouraging that it made it to phase 1 clinical trials in the first place. It passed preclinical testing, and they were putting it into human beings. And so whenever we can repurpose a drug, that saves a lot of time and effort and money and energy getting to that stage. So then in 2022, Scott's previous grad student, Rhianna Lee Ferris, published this paper where she identified that CC9 has two effects in respiratory epithelial cells. The first is that in certain CFTR variants, PTC variants, CC9 promotes read-through at the premature termination codon of particular variants and restores CFTR activity. And it simultaneously reduces ENaC activity, which is completely serendipitous finding. And she actually did the electrophysiology for this on the day that there was the storming of the Capitol. And so she was like, the world is falling apart. This is happening. And what is today? And it was a crazy experience for her. But we found that ENaC activity is reduced with this drug. So it was this completely serendipitous finding, but also really promising because basically it's making this CF airway picture look more like this non-CF airway picture, where you're restoring CFTR activity and reducing hyperactive ENaC activity. So we wanted to probe that further. She also showed in this paper that in addition to reducing ENaC activity, CC9 increases alpha ENaC mRNA in the primary cells that she was using and reduces, ENaC is a heterotrimer. It has an alpha, beta, and gamma subunit. So alpha ENaC mRNA goes up, beta and gamma ENaC mRNA go down. And then confusingly, the antibodies for the ENaC subunits aren't great, but alpha ENaC protein decreases in a dose-dependent fashion. So my grad school project then became figuring out how this works, because we had absolutely no idea. We understood more or less the mechanism by which CC9 promotes read-through at PTC variants, but we had no idea how the ENaC component of this worked. So we had a sort of three-pronged approach to this, and the first was to probe the known mechanism of action of this drug, because if they've already figured it out, we may as well take use of that, and try to figure out if that's involved in the ENaC activity reduction. So in biology, if you want to figure out how something works, break it. So we decided to knock down the two proteins that CC9 binds to see if they were necessary for the ENaC activity reduction effect that we were seeing. So cereblon is the first, and ERF3 is the second. And this is our protocol for performing CRISPR-Cas9 gene knockdowns in respiratory epithelial cells, like in ALI cultures, which I'm sure y'all are familiar with. We expand the cells, electroporate them with the guide RNAs and Cas9 that are needed for the knockdowns, expand them again, grow them at in ALI conditions, treat them with drugs, and then use our 24-channel transepithelial current clamp amplifier, or TEC24 robot, to do electrophysiology on them. And so when we knock down, let me walk you through this. So in regular cells that have no knockdown, no CRISPR, nothing like that, when we treat with DMSO, the y-axis here is a metric of ENaC activity. You see that there's a lot of it. It's a negative value, but there's a significant amount of ENaC activity here when we treat with DMSO. You'll have to trust me that this is a hashed bar, but when we treat with CC9, there's like a complete removal of ENaC activity. Then when we electroporate with scrambled guide RNAs, so there should be, in theory, no gene editing happening of any kind, that same pattern persists, where we treat with DMSO, we see ENaC activity, and when we treat with CC9, it's gone. Then we knock down ERF3A, the gene name is GSPT1 to confuse everyone, and that pattern persisted. We still see ENaC activity with DMSO, and it's pretty much gone with CC9 treatment. But then when we knock down cereblon, all of a sudden that pattern disappeared. So we still see ENaC activity even when we're treating with CC9, which is completely not what is normal. So we saw this and we're very excited, and then we replicated it in both bronchial and nasal and CF and non-CF cells. And if you look just at the orange paired bars here, it like replicated beautifully. We were super happy with this result. So we felt pretty confident in saying that this process is cereblon dependent. We need this protein here to see the ENaC activity reduction, but ERF3A independent. This protein doesn't matter. And so that led us to believe that there's some neosubstrate that is getting brought to the E3 ubiquitin ligase, ubiquitinated, and then proteasomally degraded. And in theory, that neosubstrate is probably involved in ENaC activity or regulation. So then the next question we wanted to answer was, we're assuming that this is a proteasome degrading some neosubstrate. We wanted to confirm that proteasome inhibitors would make that effect go away. And there are great small molecule proteasome inhibitors, so this was straightforward to test. So to walk you through this one again, we did a bunch of dose responsing and testing of different proteasome inhibitors before getting to this result. But when we treat again with DMSO, we see ENaC activity. When we treat with CC9, it goes away. Then when we treat with proteasome inhibitors alone, bortezomib and MG132, we did see a reduction in ENaC activity, which we didn't expect. This could just be a drug toxicity. We don't have a great explanation for it right now, but it almost looks like it's bringing us to a new baseline level of ENaC activity. Of note, proteasome inhibitors are not known to be ENaC activity reducers, so this was a surprising result, but we're just going to work with it. Then when we added CC9 on top of the proteasome inhibitors, nothing happened. So we didn't see this complete reduction in ENaC activity like we do when we treat with CC9 alone. There was no additional reduction when we added CC9 on top of the proteasome inhibitors, and that held up statistically. There's no significant difference between these and these. So here we agree that cereblon is involved. We feel pretty confident in that. We feel pretty confident that proteasomal degradation is occurring. And so the last question was to figure out, what is this neosubstrate that we haven't been able to figure out, and what is involved in ENaC activity or regulation that's getting degraded by CC9? So the first thing we did was to perform a time course of CC9 exposure. Whenever you're trying to do mass spectroscopy, you always want to make sure that you reduce both the dose and duration of exposure to a drug, because otherwise you're just going to see toxicity effects. So we found that at around 18 hours of CC9 exposure, we see the plateau at the lowest amount of ENaC activity in this particular cell type that we were testing. It's a little bit more residual ENaC activity than we saw in some other cells. But basically it plateaus at 18 hours. So that was the exposure time we wanted to use. And then we took bronchial epithelial cells from 3 non-CF donors, grew well-differentiated ALI cultures with them, treated them for 18 hours with either DMSO or CC9, and then submitted them to our mass spectroscopy core at UNC. And this was the result. So here we have the log2 fold change on the x-axis of the cells treated with either CC9 versus DMSO, and then the p-value on the y-axis. Cells in the middle basically didn't change when treated with DMSO versus CC9, no big difference. Cells on the right side of this graph were increased after CC9 treatment, and on the left they were decreased after CC9 treatment. So as you can see here, this is kind of a dirty drug. There are certainly dirtier drugs in the world, but we were hoping that there would be like one gigantic glowing target here and my life would be easy and I could graduate tomorrow. But that didn't happen. There were 251 proteins that were either above or below the biological significance cutoff, which is the absolute value of 0.6. So that's a lot. 210 of them were in this decreased after CC9 treatment category. And then 56 of them were still present if you use -1 as your significance cutoff, as a more stringent cutoff. And you saw our pipeline for CRISPR-Cas9 knockdowns. It is low throughput, so we can't knock down that many things. That said, we have certainly tried. We did a little bit of investigating, sort of trying to pick our favorite candidates based on what's known about ENaC activity and regulation, and we've tried a few and haven't found the result yet. So this is kind of a work in progress talk. But we still are working on this, like I was doing stuff in the lab this week. What we're doing right now is trying to do co-immunoprecipitation of cereblon as the bait protein and trying to capture this cloud of potential prey proteins and then analyzing that via mass spec to see if we can identify a new promising neosubstrate candidate. And then once we have that information, it's taking some optimization, we will then submit, or we'll do more CRISPR-Cas9 knockdowns and hopefully identify a target. One thing that I think is cool and interesting is that when we were looking through this, there was nothing that stood out as like an obvious, like, oh, of course, that's known to be involved in ENaC activity and regulation. We even asked ChatGPT to help us identify it, and it was like, not really, there's not much here. So we're sort of hopeful that we could be looking at a previously unexplored pathway of ENaC activity or regulation, and hopefully we'll be able to identify something that could serve as a new druggable target. This has been a group project. There are so many people to thank. Everybody in Scott's lab has been incredibly helpful, and in particular, Rihanna, Liwa, and Jacob. The proteomics core at UNC has been so helpful to us. Scott's tissue procurement and cell culture core, we could not do this work without them because they give us the cells that we use for all of these experiments. My thesis committee, our funding sources, and everybody who has donated tissues and organs or helped us get those tissues and organs that we use for this project. Thanks. No questions. Great. I will leave. Oh, maybe a question.
Thank you. That was a good talk. Thierry Vanzi, I'm from Emory and Georgia Tech. Quick question for you. So when you did your mass spec analysis of the potential targets, you saw a lot of targets. What's your take on potential, side effects, and have you tried the molecule on other cells, let's say immune cells or endothelial cells, and see whether, you might disrupt some, pretty core mechanism of protein turnover?
Yeah, that's a good question. So that's something that Scott and I talk about all the time and probably have nightmares about all the time. One of our biggest concerns is that what we could be seeing here is just a toxicity effect. I mean, clearly there's a lot of proteins that are perturbed when we treat cells with this drug, even at the lowest possible dose and the lowest possible duration. So yeah, that is something that we're trying to work out, and something, oh, it's mad at me. Do I have to turn this off? I'm just gonna leave it. What was I saying? I don't know. We're looking into doing like cell metabolomics and sort of assessing like cell toxicity assays to see if that could be a main driver. One thing that I included but didn't have time to talk about is that we also tested all of these other drugs, and I was pretty encouraged to see, this is a little bit of a complicated slide, but so this is all those other, they're called cell mods and imids, the other thalidomide derivatives. And if you look down at the benzamyl delta values here, this is 3 distinct donors. Again, we're seeing this crazy reduction in ENaC activity with CC9, but pretty much none of the other drugs are doing that. And they are all pretty toxic. I mean, I had to do a lot of dose responsing to find the doses that we could use that would both have an effect, but also not kill the cells outright. So I thought this was encouraging. But yeah, it's a great question and a concern that we have for sure. Thank you.
Hi. So CFTR is known to be important in cancer biology. So patients who have CFTR do worse mutations, but tumors can get CFTR mutations. And so it's been shown that it's not just a passenger gene, and that it actually, patients show up at higher grades of cancer, and the tumors are more metastatic, and they're more aggressive, and they're more poorly differentiated. So did CC90009 have a differential effect on patients who had tumors that also had CFTR mutations? And did anyone look?
That is such a cool question. That is news to me. I would love to know the answer. I don't know the answer, but that's a really interesting point. Yeah, I'll look into it. I don't know if they even would have that data. I don't think they've published anything from those clinical trials yet, but I check all the time. I'm very curious to know what the outcomes were like. It was a good question.
So we've made, I'm in a project with Steve Rowe and Corinne Algeli-Zafran at UAB, and we've made a ton of ERF3 degraders, or GSPT1 degraders.
Yeah.
And we see variability in the ENaC response depending upon, the different, so is it because we still have a certain constituent group that's targeted? I don't know. But the other one that has nothing to do with cereblon are the ERF1 degraders. And I got to go back and look, but they have some effect on ENaC too.
Yes, I should have put extra data in this talk, knowing that you would be in the audience, because we have tested like SRI413 and 5 on these cells, and I also can't remember off the top of. I don't think we saw ENaC activity reduction except at very, very high doses that were like toxic to the cells. But I would have to go back and check as well. But it's an interesting question too. Like, could it be some sort of ERF3 related thing, even though when we do our knockdowns, we're not seeing ENaC activity reduction, or we still do see it? I don't know.
Just 90009, is it just ERF3 degraders or fermentation?
Right, yeah, lots of questions about this. Yeah. Cool. Thank you.
All right, thanks everybody. So our second speaker is Isabella Sermet-Gaudelus from the French National Institute.
Thank you. Thank you very much to have invited me to talk about novel readthrough compounds for nonsense mutation that we are currently developing in the lab. So my disclosure, I'm a member of the scientific board of ENaC Therapeutics, and I'm also PI of Center enrolling patients in Vertex Pharmaceuticals trials. There are currently nearly 200 PTC mutation in the CF mutation database at CKITS, the most prevalent being G542X, about 5% of people with CF, followed by W1282X, about 2.5% with CF, and then R553X, R1162X, and E60X mutations. These top 5 are UGA PTC, apart E60X, which is a UAG. Patients carrying 2 PTC have a more severe phenotypes, as illustrated here in this cohort of more than 26,000 European people with CF. In Europe, the patients carrying 2 PTC here in blue show more rapid decline in respiratory function from childhood, resulting in lower FEV1 in adulthood as compared to the delta 508 homozygous patients here in black and the cohorts of PTC delta 508 compound heterozygotes. So there is a high unmet need for these patients as they are not eligible for CFTR modulators. A nonsense mutations introduces a premature stop codon in mRNA, UAG, UGA, or UAA that induces mRNA degradation by the nonsense-mediated decay, which is activated during the pioneer round of translation. And for those transcripts which escape NMD degradation, PTC induces a premature translation termination and release of the truncated protein, which is most of the time not functional. Release factors recognize stop codon and initiate termination. Translation read-through involves mispairing of a stop codon of a PTC with a near-cognate RNA rather than its termination factor. This physiologically actually occurs at a rate ranging between 0.001% and 0.1%, but it can be pharmacologically induced at a higher rate. Different read-through strategies are currently being developed, such as engineered tRNA or antisense oligonucleotides, but today I'm going to focus on read-through molecules. Many factors may influence PTC read-through, including efficiency of NMD, which can be inhibited by NMD inhibitor, the PTC itself, UGA being more permissive to read-through, the nucleotide context surrounding the PTC, and also the recoded amino acids, because the neoprotein formed can be correctable by CFTR modulators. The potential of aminoglycosides to read-through disease causing PTC was investigated more than 10 years ago for gentamicin by the group of David Bedwell, among others, leading to also to modified synthetic aminoglycosides, less toxic than gentamicin. ELOC02 showed good read-through in vitro, but no improvement in respiratory function in a pilot clinical trials, and this will be the reference molecules for our studies. This failure paved the way for high-throughput strategies, leading to the discovery of PTC-124 Altalarend, which did not display toxicity, but showed no clinical efficacy in different clinical trials, and more recently, adenine analogs such as the aminopurine, DIP, and more recently, release factors degraders by the group of David Bedwell and the CF Foundations, and the group of Scott Randall, compounds which reduce the abundance of the termination factors, RF1 or RF3, and thus increase the probability of near-cognate RNA accommodation at PTC. They have shown activity in vitro, but also display some toxicity in vitro. We took a completely different approach, showing a strategy of structure-based design. This was based on the work of the group of Marat Yusupov here in Strasbourg in France, and the collaboration with Orania Therapeutics, which is the spin-off from their lab. This group was the first to describe the crystal structure of a whole bacterial ribosome in associations, which is functional ligand, mRNA and the tRNA at the A, E, and the P sites. And then 10 years later, they described at the atomic resolution the crystal structure of a whole eukaryotic ribosome. This enabled them to solve the crystal structure of the eukaryotic ribosome in complex with various inhibitors to understand the molecular mechanism of action of these drugs. These studies constitute a major step forward, and this enabled the group more recently to determine the mechanism of action of mRNA reading frame maintenance during eukaryotic ribosomal translocation. And here is shown from one of the latest publication of the group, the crystal structure of the whole eukaryotic ribosome in a translocation state containing mRNA here in orange, tRNA here at the E and the P site, and the elongation factor, EF2, acting as a pole to maintain the directionality of the translation. Here you can see here the ribosomal RNA and the ribosomal protein here in cyan and dark blue. Here a more focused view of the decoding center obtained here again from the X-ray crystal structure, where you can observe here the decoding center in the small subunit interfacing with the large subunit and contacting with the mRNA exposed codon here in orange, base pairing with the tRNA anticodon. Importantly, during the base pairing, highly conserved adenine here, locating in the ribosomal helix 44, flip out to stabilize the codon-anticodon pair. And this is a feature which is absolutely crucial, to discriminate between Watson and Crick base pairing and mismatch for proper elongation. Importantly, read-through molecules here in yellow can bind to the decoding center and force this ripped-out conformation, therefore stabilizing the pairing of the PTC with the near-cognate RNA and allowing, therefore, to continue the elongation. And the structure-based drug design approach is based on the crystal structure of the complex of the ribosome with the mRNA, tRNA, elongation factor, and the compound itself. Here you can see the experimental electron density, which is obtained here from the eukaryotic ribosomal crystals, here studied by X-ray diffraction, and here placing the atomic structure in this electron density will enable to design the structure of new compounds which would increase the affinity of the A site with near-cognate tRNA. Then these new structure are then tested in readthrough assay, first TNT cell-free system, and then anti-proliferative assay using cells expressing P53. This leads to drug candidates, and we chose 2 main drug candidates, which I'm going to call URN1 for the first generation and URN2 for the second generation. We first tested them in different CFTR PTC contexts using a dual reporter system in LR cells expressing the PTC and its 9 nucleotides on either sides in comparison to an in-frame control structure. So this was already published a few years ago. So as you can see here, comparing LOX02 here in blue, URN1 here in green, and URN2 in ochre, URN1 and 2 is more efficient than LOX02 for UGA, UAG, and UA readthrough. And this is obtained and observed in variants, in different variants linked to UGA, G542X, S1196X, and W1282X. And also for a mutation associated with the UAG, W846X, and UAA Y122X. So we tested in cellular system the readthrough efficiency, and we first tested that in the 16-HBE cells gene edited from the CF Foundation. So in the next slides, I'm going to show the readthrough efficiency as a readthrough in CFTR-dependent chloride transport in using chamber experiment expressed as a percentage of the wild type in different conditions, testing the drug at 100 micromolar and 200 micromolar here in combination with ETI here and ETI and NMD inhibitor. So as you can observe here for URN1, which is the first generation compound, there is a consistent 2-fold increase versus LOX02 versus as compared here at 100 micromolar and at 200 micromolar, reaching 4.6% of the wild type maximal readthrough efficiency with in combination at 200 micromolar with ETI and MD inhibitor. URN2 second generation compound was much more efficient, reaching a 4-fold increase versus LOX02 at 100 micromolar here, as you can observe, and 8-fold increase at 200 micromolar, reaching up to 10% of the wild type in combination with ETI. So a consistent improvement of readthrough efficiency with URN1 and URN2, but URN2 being consistently more efficient. So in the next slides, I'm going to focus on the experiments using URN2. So this rescue in CFTR activity was associated with rescue in CFTR expression, and you can observe here the nice restoration of the C mature band B here in the 16-HB incubated with URN2 at 200 micromolar with a slight increase with the association with the NMD inhibitor. And this is also supported by the increase in the transcript level as compared to LX02 here at 100 micromolar and 200 micromolar. And here is a summary of the different Western blot experiments that we are currently doing with a nice rescue up to 20% of the wild type of URN2 of C-band with URN2, a slight increase by the association with ETI and ND inhibitor, and a slight rescue of the mature band with the ND inhibitor itself. So these results in cell lines were also confirmed in results in primary cells. Here is shown results in primary cell from a G542X inhibitor patient, with a consistent improvement of URN2, 2-fold increase in readthrough versus LOX02 at the different concentration, as you can see here, and a rescue of a maximal activity up to 30% of the wild type when URN2 was combined with ETI and ND inhibitor, and 18% when URN2 was combined with ETI. This was also observed in human rectal organoids of a G542X homozygotes patient, 2-fold increase here at the different concentration with URN2 versus LOX02, and a maximal activity of 35% when URN2 was combined with ETI and NDI, and 32% with ETI only. So we are now testing other variants linked to UGA, PTC. Here I show the results of R553X homozygous nasal cells, where you can observe here absolutely no efficiency of read-through of LOX02, small efficiency, I should say, for urine2, but much less than that observed with G542X, probably because of a very high activity of NMD, as shown here as unraveled by the efficiently rescue when we combined urine2 with NMD inhibitor. We also tested other mutants linked to UAA, Y122X here, and where we showed also that urine2 increased by threefold the read-through efficiency as compared to LOX02 at the 2 concentration, leading to a maximal activity to 10% read-through of the Y type when urine2 was combined with ETI and NMD inhibitor. And this was also, these results obtained in cell lines was confirmed in primary nasal cells of homozygous patient, where you can observe here a consistent twofold increase at the 2 concentration versus LOX02 here, and a maximal activity of 25% of the normal when urine2 was combined with ETI and NMD inhibitor. And we are currently investigating that also in rectal organoids, and where you can see here that urine2 combined with ETI at 200 micromolar reaches more or less 17% of the Y-type activity. We are currently also investigating mutations linked to UAG. And here is the first result with E60X, where you can observe that urine2 at 100 micromolar reaches 6% of the activity, but I cannot show here at increased concentration, and also the combination with ETI, because the patient carried in trans a mutation which was correctable by ETI. So in summary, we have a consistent read-through efficiency for urine2 in 16BE gene-edited cell lines, urine2 being more potent than urine1 and LOX02, up to 10% of the CFTR normal activity when it was combined with ETI here, and with ETI and MD inhibitor for Y122X, and very importantly, in nasal cells also the same profile in G542X, R553X, Y122X, and E60X mutations, up to 18% of the CFTR normal activity with ETI for G542X and Y122X here, but only 5% for R553X, because probably of a very high activity of NMD in these nasal cells. Importantly, we did not observe apparent toxicity in primary airway culture, as shown here by no change in the transepithelial resistance at the different concentration, no also increase in LDH activity, and we are now finishing MTT. And we finally did not observe here any natural stop codon read-through based on genome-wide ribosome profiling experiments in LR cells, as you can see here, when compared to untreated, which was not the case for G541H, as published by our collaborator. So in conclusion, better understanding of read-through compound mechanism, low rational design of new effective compounds. We demonstrate read-through efficiency for urine2, more potent than urine1 in 3 different systems, respiratory cell lines, primary nasal cells, and rectal organoids. Urine2 is effective for all 3 PTC, UAG, UAA, and UGA, can rescue up to 20% of CFTR activity in combination with ETI for G542X and Y122X, with a high activity of NMD in all these cells, and mainly for R553X, with no apparent toxicity. Work ongoing in future direction with urine2. We are currently investigating other genotypes. We aim to identify the amino acid recoded, the combination of urine2 with other read-through strategies, ERF1 degrader. We aim to assess the effect on normal termination and cellular transcriptome in more relevant cell lines than the LR cells, and mainly now go to administration in vivo to the G542X mouse. URN2 molecules can potentially treat many diseases, and there is also ongoing studies in epidermolysis bullosa from now. And with this, I want to really thank all the people in the lab working on this project, and also all our collaborators, Olivier Namy at Orsay, Marat Yousupov Group, and Jean-Paul Renaud at Urania Therapeutics. And with this, I thank you.
Wow, I don't think you took a breath the whole talk. That was a lot of data. Very nice data, too. So, Yousupov, so are these drugs targeting the decoding site, helix 44? That's kind of what you implied, but you didn't say that. You know, with the 1745, 1746 flipping out. So are they aminoglycosides? Because that's where aminoglycosides bind. Or are they something completely chemically different?
They're related to aminoglycosides. That's what I can say.
So that's, people call ELX.
Oh, they are derived from aminoglycosides. To tell it more, they are derived from aminoglycoside structure. To tell it more accurately.
So it's kind of like a ELX02 type thing with different chemical moieties. Okay. So yeah, that's very nice work.
Thank you.
There's a question online in the chat is, do you expect similar results with W1128-2X?
So we are currently investigating that, and there is results for W1282X as well. Less than G542X, that's what I can say, but we need to explore more primary nasal cells from patients.
Thanks again. Really nice talk. When you showed data suggesting selectivity for the CFTR stop codon versus natural stop codon read through. And I guess I'm wondering, biophysically, how do you rationalize that? Where would that selectivity come from, and can it be rationally tuned in to optimize?
So I think that I'm not the good person to answer that. I think that David Bedwell could answer better than me. So there is some rationale, yes, because the structure of the ribosome is conformed to read through the PTC more than the natural terminal. So you can answer better than me, because actually I'm not.
So there's stuff that binds to the 3' UTR that in some cases have been shown to interact with ERF3 and stimulate termination at normal stop codons. And when you have a premature stop that's way far away, that interaction doesn't occur. So the hand waving argument is that promotes more read through at PTCs. And while I'm up here, let me ask one more question.
Yeah, thank you to save me.
So the devil in the details with the aminoglycosides are nephrotoxicity and ototoxicity. So can you say anything about that?
No.
I think with ELX, they're better, but in the clinical trials, they couldn't go beyond a certain point, and that wasn't good enough to get suppression of CFTR stop.
So absolutely, that's a good question. Now we need to go to animal studies. We are currently now implementing in wild type mouse, but we absolutely need fundings to explore that in G542X humanized mouse. That's a project that we aim for now, just to really know the toxicity of these drugs.
Mice are pretty resistant to aminoglycoside.
Yes, I know. Yeah, I know, but so we need fundings to go after these studies in animals to go to humans, absolutely.
All right, well, thank you. So our next talk is from Zitong Wang, who's a PhD student at the University of Michigan, who's going to talk to us about pulmonary delivery of circular RNA. Thank you.
Okay, so good morning, everyone. My name is Zitong Wang. I'm a third-year PhD student from Dr. Julian Zhu's lab at the College of Pharmacy, Department of Pharmaceutical Science at the University of Michigan. So today I'm titled Pulmonary Delivery of Circular RNA Therapeutics Using Pulmonary Surfactant Incorporated Lipid Nanoparticles for Cystic Fibrosis Therapy. This is my disclosure slide. I have no relationships to disclose related to this presentation. So as we know, cystic fibrosis is caused by many different CFTR mutations, which encoding the CF transmembrane conductance regulatory protein. As we see in this pie chart, the most common CF mutation is F508del, and the other including G542X, G551D, etc. The CFTR mutations can be further classified into 6 types. In class 1, there is no protein produced, which could be due to early protein production termination, or there's a large chunk of DNA mutation. In class 2, there's defective trafficking in the CFTR protein. The F508del is one of the example of the class 2 mutation. And class 3, there's no function protein on the surface. Class 4, there's less function protein on the surface. 5 and 6, there's either less protein or less stable protein on the cell surface. Here we see the current CFTR modulators targeting specific CFTR mutations, including correctors, potentiators, stabilizers, and amplifiers. While these modulators are very effective, there are types of specific mutations cannot be treated by these modulators. According to the literature, there are about 8% of people with CF are not very responsive to the current modulator therapy. Here, where the gene therapy and RNA therapy come in. So, nucleus therapy, including gene therapy, delivering used adeno-associated virus, AAV, or lentivirus vectors. There's also RNA replace therapy, mRNA-based therapies. There's 3 clinical trials to restore CFTR expression. There's antisense oligonucleotides targeting specific mutations of CFTR. And there are also emerging platforms, including CRISPR-Cas9, etc. So our lab are specifically interested in RNA therapy, mRNA therapy, and circular RNA therapy. Which due to its less immunogenicity. However, based on our group's past work, a critical limitation of this current mRNA therapy are their transient protein expression, which necessitates their frequent dosing, which could be further induce the tolerability issue. So our group past work has shown that our circular RNA shows more stability than the mRNA linear version in the storage of -20. And also on the right-hand side, there's prolonged protein expression for the circular RNA over the linear counterpart. So based on that, we have designed a circular RNA encoding CFTR protein generated from in vitro transcription IVT by PIE1 method, which is a permuted group 1 intron exon reaction, which basically relies on the autocatalytic activity of the ribosome within the sequence. And figure B, we show that this is gel electrophoresis data. The samples are loaded in duplicate. The left 2 lanes are circularization product of the IVT reaction. The right 2 lanes are circularization product plus RNA treatment, which basically removes everything byproduct from the IVT reaction. And further, we confirm through the RT-PCR experiments that we basically designed 2 primers flanking through the ligation sites, and to further confirm that this circular RNA is actually being circularized. And next, we move to study the stability of our circular RNA. We see in the panel NB, after 7 days of storage, the circular RNA shows enhanced stability over the mRNA linear counterpart. And again, in the FBS condition, with increasing concentration of FBS, the circular RNA still shows better stability over the linear mRNA version. So in brief, this slide shows us the circular RNA is highly thermostable and biostable. And next step, we took this circular RNA and transfect them into cells. We see that after transfection into the cells, over a 7 days period, the circular RNA still shows better transfection efficiency over the mRNA version. This was transfected by lipofectomy. And then we did a using chamber study to study whether this transfection can restore the channel restoration in F508 DEL cells. So we see that if we add the phospholipid and GlyH101, which are CFTR activator and inhibitor respectively, the current change for these cells for the circular RNA is significantly higher than the MRI version. So basically it demonstrates that circular RNA is doing better job for the channel restoration function. However, while these in vitro studies are pretty promising, the next major hurdle we encountered is to deliver the RNA into the LV epithelial cells. We know we want to deliver the RNA system into the conducting zone. We know that LV epithelials are lined with very thick mucus, especially in CF patients. Their mucus are much more thicker. So in order to solve that, our lab has developed a pulmonary surfactant coated lipid nanoparticles, which basically have a regular LNP core, and then you coat that with the pulmonary surfactant outer layer, and our circular RNA are within this LNP core. And shown in the cryo-EM image, our LNP shows pretty spherical morphology, and these diameter are about 120 in diameter. And basically the pulmonary surfactant would just coat it outside of the regular RNP. And next study, we move into in vivo. We using this PS coated nanoparticles, I abbreviate as PSLNP, to inject the mice by intratracheal injection. We give the mice with a circular luciferase RNA, and we see that over time, the PSLNP has more protein expression, RNA expression, than the regular RNP. And then we want to test the mechanism behind that. This is one of our mechanistic study. So we, in collaboration with Dr. Bruno Lab in Pharmacy at the University of Michigan, so we have this LIHBE cells, which they spontaneously secrete the mucus on the apical site. So we give this LIHBE model with the EGFE mRNA either in regular RNP or PSLNP. So, and after a designated time, we took all of the cells and analyzed their signal. So we see here there's more protein being taken up and expressed for the PSLNP over the regular RNP. So briefly, this slide shows that the PSLNP can promote RNA expression and protein expression than the regular RNP. Then we use this PSLNPs to study in vivo. We use the CFKO mice from Jackson Lab, and we run a Western blot. There are three experimental groups: the mRNA version, circular RNA, and the PS circular RNA. And we see from this in vivo study in the CF mice, the PSLNP shows higher CFTR expression over the circular RNA and over the mRNA version. And next, we study we want to evaluate whether this PSLNP can restore the CFTR function. We, in collaboration with Dr. Jack Shu Lab in Michigan Medicine, we have a CF508 DEL rapid model. So we use our LNP to give this CFTR mutated rapid model to test their nasal potential difference. Abbreviated as NPD. So briefly, you would just put one of the electrode under the rabbit's nostril, and you just record the PD response flowing through different solutions. And here is an example for flowing through phospholipid. So we see the blue dot on the left-hand side, the wet type and the CF plus ETI treatment. The PD response is about like -17. And then the day 0 shows the CF rabbit at the initial time point. It's about like -3 or 4 or something. And then with our single dose of circular RNA, we see that the CF rabbit shows a PD response, shows a gradual increase and peak at the day 3 and continuously about day 7. And and this is an ongoing study. We need to do more study on this rabbit. So with that, conclusion is our circular RNA CFTR can prolong CFTR expression and functional restoration. And our PSLNPs were optimized for this pulmonary delivery of circular RNA, and it shows efficient and long-lasting CFTR expression in both cells and CFKO mice model. And in the rabbit study, our PSLNP in the single dose can restore chloride channel function in the CF508 rabbits, and it's still an ongoing study. with that, I'd like to thank you all your time and acknowledge all of my lab members and our collaborators, and as well as the extra funding. Yep. Thank you. And ensure space without poster number 273.
Hi, very nice presentation, David Geller from Arcturus. I was just curious as to what kind of surfactant you were using, whether it was synthetic or animal-derived, or commercially available, or if you can say.
I will leave that question to my PI, so please contact him. Yeah.
Great talk. Do you have any data on the immunogenicity of the circularized RNA versus its non-circularized counterpart?
Okay, so I have a data but not shown here. So the circular RNA, if there's because current mRNA version is typically modified with pseudouridine or something else. So I can say as the circular RNA is much less immunogenicity than the non-modified RNA, and it shows less immunogenicity than pseudo-modified mRNA.
Hi, really nice talk to you, Ramadian from Emory and Georgia Tech. A quick question for you regarding your rabbit data.
Yeah, sure.
You were showing essentially a lag in response, right? After administration, and then you show the response was kind of therapeutically effective or in the right region that you were aiming for about 3 days. So does that kind of jive well with your in vitro data in terms of the expression of the circular RNA and then the stability? Do you have equivalent data in your rabbit?
So you're asking our rabbit data, the circular RNA peak at day 3, is there corresponding data for the in vitro study?
Yeah, I'm basically just questioning, in terms of what you showed, the lag in terms of the expression and then the, the stability over time.
Yeah, that's a very great question. So in vitro, there is actually a lag for the circular RNA expression over the mRNA. So mRNA typically peak at 24 hours, and circular RNA will just maybe peak at 3 or 4 days and then last longer than the mRNA. So that's what I see in the in vitro study.
And then is the decay of the circular RNA dependent upon the actual transcription? That is, as soon as it's being engaged?
Yeah.
Then it is kind of marked for degradation?
No, there's, according to the literature, the degrading mechanism for the circular RNA are different than the linear version. You not just, so I don't believe that there's be quite quickly degradation mechanism for a circular RNA, but that's a major future perspective that we will study.
Thank you.
I have a question. In the in vivo dosing, have you done repeated dosing over time?
No, not really. And that's also a future study because the rabbit are very rare. So we're waiting for the rabbit so we can do the repeated dosing for this treatment. Yeah. Thank you for your question.
If there's no further questions, let's thank our speaker.
Thank you.
All right. Our next speaker is Naomi Celise from University of Illinois, and is going to talk to us about molecular prosthetics restore anion movement in CF intestinal models.
Thank you for the introduction, and I have no disclosures to disclose. So to begin, I want to define a molecular prosthetic, because in Berkeley Laboratories, we define a molecular prosthetic as a small molecule that can restore or replace the function of a protein that's defective, such as that of CFTR and cystic fibrosis. And we saw the opportunity with the natural product amphotericin B, since it has channel-forming properties, to first ask whether it can mitigate the decrease in airway surface liquid pH, the increase in airway surface liquid viscosity, and the decrease in antibacterial activity that's commonly observed in the cystic fibrosis airway by restoring anion movement. And my former colleague Katrina, in collaboration with Walsh Laboratories, began investigating this utilizing primary airway epithelia derived from the lungs of people with cystic fibrosis of various mutation types. So beginning with airway surface liquid pH, as shown here on the y-axis. On the x-axis, I'm showing the different conditions that were tested with amphotericin B, either here in the yellow, or without amphotericin B. And just to note, amphotericin B will be abbreviated as A and B throughout this presentation. We can see here that upon incubating these primary cultures with amphotericin B, there's an increase in airway surface liquid pH. Now, if we look at airway surface liquid viscosity, addition of amphotericin B also decreased the viscosity. And these two effects led to an overall increase in antibacterial activity, as you can see here by the increased killing of the bacteria Staphylococcus aureus. And these results, along with others, led to a formulation of amphotericin B that entered clinical trials. And if you want to learn more about these clinical trial results, please visit poster 867 tomorrow. Because the focus of this presentation will be to take a first look at molecular prosthetics and cystic fibrosis intestinal epithelia, where we ask whether anion movement could be restored with our molecular prosthetics, and where we also ask if a second-generation molecular prosthetic, known as amphotericin B-AA, could do a better job at restoring anion movement. The reason why is because amphotericin B-AA is a variant of amphotericin B that was found by my colleagues, Jonathan Marin-Toledo and Diana Greenan, to have an increased permeability. The first part of our investigation could not be possible without our collaborators at Iowa University, one who is Professor Ian Thornell, and without the help of my colleague Diana Greenan. So the purpose of these studies at Iowa University was to utilize intestinal segments derived from pig duodenum and inject them with our molecular prosthetic to ask whether small molecule ion channels could permeate this tissue and result in a restoration of anion movement, which we quantified as increases of intestinal segment weight due to the movement of water into the lumen. But more specifically, shortly after birth, pig litters were sacrificed of the non-cystic fibrosis and cystic fibrosis genotype. We obtained the duodenum. I then washed it, segmented it, and sutured it as shown here. And with the microneedle, I either injected amphotericin B, amphotericin B-AA, saline, or IBMX forskolin. I then placed these intestinal segments into a ringer bath solution that was bubbled with 5% CO2 and maintained at 37 degrees Celsius, where me and my colleague Diana Greenan took weight measurements of these intestinal segments every 10 minutes. And here are the results for this study, beginning with the non-cystic fibrosis genotype. On the y-axis, representing change of volume normalized to intestinal segment length. On the x-axis, we have the results for our saline vehicle control in gray, and in red for the intestinal segments that were injected with IBMX forskolin. As you can see here, addition of IBMX forskolin led to increases of fluid secretion into this intestinal segment lumen, comparative to our saline control. And this is as expected since CFTR is present on the cell membrane and is activated by IBMX and forskolin. Now looking at the cystic fibrosis genotype. Here, in the absence of CFTR, addition of IBMX forskolin did not lead to any changes relative to our vehicle, which is why it was promising to see that addition of our molecular prosthetics, whether it be amphotericin B, shown here in the yellow, or amphotericin B-AA, shown here in the blue, not only led to increases in intestinal fluid secretion that were not significantly different from that of IBMX forskolin for the non-cystic fibrosis genotype, but also led to increases for a cystic fibrosis genotype, as shown here. And preliminary studies in which we utilize a voltage meter and microelectrode to measure the change of voltage upon injection with either our vehicle, amphotericin B, amphotericin B-AA, showed that there was voltage changes observed in our groups relative to that of vehicle for the three conditions tested here, which suggests that these increases in fluid secretion were either due to CFTR activation or successful small molecule ion channel formation. Now, to more quantitatively assess anion movement through our small molecule ion channels, We utilized a human intestinal cell line, CACO2, with CFTR knocked out, and radiolabeled bicarbonate. For this experiment, I incubated CACO2 CFTR knockout epithelia for 48 hours with, in this case, amphotericin B in the luminal surface liquid, which is abbreviated as LSL. And following the 48-hour incubation period, I then acutely added radiolabeled bicarbonate onto the basolateral side. And after 30-minute incubation time, I collected the luminal surface liquid to quantify how much bicarbonate moved from the basolateral side through our small molecule ion channel to the luminal surface liquid through a scintillation counter. That's what's represented here on the y-axis. And on the x-axis, I'm showing our results for the cystic fibrosis group without any small molecule addition, and the range of concentrations that were tested for amphotericin B that are from 2 to 100 micromolar, represented here by the yellow. We can see that relative to our cystic fibrosis group without any small molecule, there was an increase in bicarbonate secretion to the luminal surface liquid, which is about 50% of that of wild-type CFTR. Now I repeated this experiment, but this time with amphotericin B-AA. And not surprisingly, at the range of concentrations tested in this assay, ranging from 2 to 100 micromolar of amphotericin B-AA, we also observed an increase of bicarbonate secretion to the luminal surface liquid. We did a head-to-head comparison to assess how bicarbonate secretion moved through amphotericin B base channels compared to our more anion-permeable small molecule. As you can see here, the increased anion permeability of amphotericin B-AA base channels led to a further increase of bicarbonate secretion to the luminal surface liquid of these CACO2 CFTR knockout epithelia. And to evaluate the effect on overall pH of our molecular prosthetics, I replaced the ringer solution of the CACO2 CFTR knockout epithelia, or the media solution, with a ringer solution that was bicarbonate and potassium free, and I made it slightly acidic to a pH of 5. I then either incubated these cultures with either our vehicle, amphotericin B, or amphotericin B-AA for 48 hours. The basolateral solution did contain bicarbonate and was pH to 7.4. And before I get to these results for this experiment, I want to add some context behind our slide title, which is a divergent dose response for amphotericin B, by showing some previously obtained data with airway epithelia with my colleague Jonathan Marin-Toledo, specifically for the CUFI1 cell line that's homozygous for the delta F508 mutation, in which we measured the change in airway surface liquid pH, as shown here on the y-axis, at different concentrations tested for either amphotericin B in the yellow or amphotericin B-AA in the blue. And we saw that compared to our vehicle, at 2 micromolar, there was a change in airway surface liquid pH for both small molecules. But as you can see here, it was more efficacious for amphotericin B-AA. And this increase was sustained throughout the high concentrations tested. However, if you look at amphotericin B, as we increased the concentration of amphotericin B, the airway surface liquid pH began to decrease until it reached baseline levels. Now looking at the intestinal epithelia results with the CACO2 CFTR knockout. Now looking at change of luminal surface liquid pH on the y-axis, and again on the x-axis is a range of concentrations that we tested. As we saw before, at 2 micromolar concentrations, there was a change in luminal surface liquid pH relative to vehicle that was more efficacious for amphotericin B-AA compared to amphotericin B, but in contrast to what we observed in the airway, as the concentration of amphotericin B increased, we did not see a drop in the pH. And this led us to investigate what the loss of efficacy of amphotericin B at high concentrations could be tied to. And what we found for results in our airway epithelial cell line was that at high amphotericin B concentrations, there was too much cation movement to the airway surface liquid due to amphotericin B increased permeability for cations over anions. And as a result, this led to an increase of activity and expression of the proton-potassium exchanger ATP12A. We therefore asked if in our CACO2 cell line, ATP12A activity and expression was being left unaffected. And we first looked at the activity with a protocol that was adopted from Bulik-Sullivan, Thornell, and other authors, in which we, as before, incubated our cell line, CACO2 CFTR knockout, with amphotericin B at 50 micromolars or with our vehicle for 48 hours. Following the 48-hour period, I replaced the culture medium with a ringer solution that was bicarbonate-free. And just to note, the basolateral solution was also bicarbonate-free. And to the apical solution, it either contained 1 millimolar of wallbane or did not contain any wallbane. And wallbane is an inhibitor of ATP12A. So under these conditions, we're able to assess the change of pH that is sensitive to ATP12A inhibition. And this is plotted as proton flux over a period of time. And as you can see here, there was no changes in proton flux observed under the amphotericin B at 50 micromolar conditions or a vehicle that were sensitive to ATP12A inhibition. We also did not observe any changes in ATP12A protein expression through Western blot analysis of amphotericin B at 50 micromolar relative to our vehicle, which contrasts the results that we observed in the cystic fibrosis airway. Therefore, we further asked if there was an inherent difference in ATP12A expression in the CACO2 CFTR knockout epithelia relative to our CUFI1 cell line. To do this, I utilized RT-qPCR to look at ATP12A mRNA expression and immunofluorescence to also look at ATP12A protein expression. As you can see here, in both cases, the CACO2 CFTR knockout cell line has a decreased expression of ATP12A mRNA and ATP12A protein relative to that of our CUFI1 cell line. So overall, I hope I helped demonstrate that molecular prosthetics are a promising approach to help restore anion movement to cystic fibrosis airway epithelia and cystic fibrosis intestinal epithelia. And that, like traditional drug design, we can optimize our lead molecule to have a better functional restoration, as demonstrated with amphotericin B-AA. At the same time, we're still learning about the interface of our molecular prosthetics with endogenous proteins and their effects on cellular biology. So thank you all for your time and attention. And of course, thank you to everyone who has been involved in this project and to all the funding sources. And I'll be happy to answer any questions.
You suggested that the new amphotericin AA had a better anion selectivity. You also noted that amphotericin has a cation, potassium. So what is, for amphotericin, have you measured the PK to P anion, the permeability of the anion to the permeability of the potassium? And
yes.
How does that number change in your modified prosthetic?
Yes, so that's actually work that was led by my two colleagues. And it's the PCL over PNA for amphotericin B is around 0.03, and the increased selectivity for amphotericin B AA is 100 times that. And that work can be found currently right now. It's under revision, but it could be found in the bioarchive. It just goes into a lot more nice detail about that.
Yeah, so I'm Beate, just asking you one question about the amphotericin B. So it's known to promobilize both apical and basolateral cell membranes. And I was wondering, have you looked into some toxic effects towards the basolateral membrane? So when you incubate with 200 micromolar, I'm just a little bit concerned that it may cross over to the basolateral membrane. Can you comment on that, please?
Yes, so with amphotericin B, we have done a formulation of amphotericin B did enter clinical trials and has gone to extensive tox studies. We have not seen any talks related to the administration of amphotericin B in people. And in epithelia cells themselves, we did do toxicity studies, but only to the airway, and did not observe toxic concerns up to the concentrations tested. We did see some toxicity at 100 micromolar. Mm-hmm.
Very nice. So you showed data with intestinal loops and with CACO2s. have you tried rectal organoids or rectal tissue grown as 2D epithelia from people with cystic fibrosis?
No, but that's definitely something that is going to or should be done for the next phase of this project.
Well, all the best with those studies.
Thank you.
All right, well, let's thank our speaker. So our final talk is Inera Manzarez-Fornes, who's a PhD student. She's going to be talking to us about restoration of anion transport to the W12882X.
Good morning, all. I would like to begin by thanking this opportunity to Professor Brook and Professor Long. I would like to start with the aim of my studies, which is channel replacement for cystic fibrosis using artificial anion transporters. CFTR is made in the ER, then matured in the Golgi, and then transported to the apical membrane, where it acts as an ion channel. For nonsense CFTR variants, such as W1282X, there is an introduction of a premature termination codon into the CFTR mRNA. This leads to a degradation of the mRNA by the nonsense-mediated decay. Therefore, no CFTR protein is produced, and there are no functional channels in the apical membrane. Then, one pharmacological approach is to pharmacologically rescue CFTR using a combination of drugs. This first using an inhibiting NMD, second correcting CFTR production, and then potentiating CFTR function. Then by combining the experimental NMD inhibitor SMG1I, Venturini et al. Restored 34% of CFTR function in 16HB14O- cells expressing W1282X. An alternative approach is to use channel replacement for CFTR for CF epithelia using these artificial anion transporters, or termed anionophores. These are small molecules that bind an anion on one side of the membrane, then transport it across the membrane and release it on the other side. This anionophore has an anion binding pocket and hydrophobic scaffold that enhances this deliverability to the cells. Our lead family are the decalins here on the right side, and then but my studies are going to focus on difluoroorthophenyl bisureas, termed DIF-OPBU anionophores, which are easier to synthesize. Thus, we want to investigate whether these DIF-OPBU anionophores alone and combined with NMD inhibition and CFTR modulation restore anion transport to W1282X CFTR expressing human broccoli epithelial cells. Here are the 7 compounds that I studied. The urea terminals were modified first by introducing phenyl groups, which enhances rigidity, lipophilicity, and membrane affinity. Second, by introducing different levels of fluorination, here and here, which enhances anion binding. Then, to test this anion transport, we use halide-sensitive yellow fluorescent protein in cells. Then we wanted to investigate first the acute effect of DIF-OPBU anionophores on their own. We use, to screen the anionophores, we use Fischer-Rath thyroid cells expressing yellow fluorescent protein alone. Then I studied anion transport of these compounds using the 16HB14O-, co-expressing W1282X CFTR, and the yellow fluorescent protein. Then W1282X CFTR HBYFP cells. I incubated the cells with 50 micromolar of DIF-OPBU and ionophores at 37 degrees for 10 minutes. In this case, when the cells were bathed in chloride, the fluorescence was high. Then, when iodide was added, the fluorescence was reduced. The iodide was only transported inside the cells by the anion transport, the ionophores. Therefore, this measure of fluorescent quenching relates to the anion transport by an ionophores. Here we have a summary of the acute effects of these compounds. On the left-hand side, we have the rate of anion transport, and here, the larger the negative value, the greater the transport activity. The anion transport activity of compound 157 was similar to our reference decaling compound 11. By contrast, compound 158 had double activity than our reference compound decaling 11. The rest of the ionophores had little or very little anion transport activity. Then, these modifications on the structure by adding high level of fluorination and phenyl group to increase the rigidity clearly enhance anion activity, as we can see in 158. We selected 157, 158, 159, and 160 compounds to be tested in W1282X CFTR HBYFP cells. Here, we can see a huge increase on activity for 159 and 160, but still the anion transporter with the highest anion transport activity was 158. Thus, the anion transport activity is cell type dependent. Then we wanted to see how was the chronic effect of treating the cells with 10 micromolar of DIF-OPBU on their own at 37 degrees for 24 hours. And also, we wanted to see the synergistic effect between DIF-OPBU and ionophores and SMG1I, NMD inhibitor, and CFTR modulation, LS-Captor, TS-Captor, IVA-Captor. Thus, I will call it, for the rest of the presentation, SETI. Then, for that, we incubated also the cells at 37 degrees for 24 hours, and just prior to the iodide addition, stimulated the rescue W1282X CFTR with forskolin. Then again, the anion transport was measured by the iodide-induced fluorescent quenching. On the left side, we have representative time courses of iodide-induced fluorescent quenching of the 4 ionophores. On the right side, we have a summary of the anion transport rate of the ionophores. And as before, as the acute treatment, 158 was the compound with the highest anion transport activity, followed by 157 and 159, with 160 with less activity. Here we have the chronic effect of the ionophores combined in the presence of SETI, and then stimulated by forskolin before iodide addition. On the right side, we have the summary data that shows some synergistic effect between 158, 159, and 160, which is going to be more clearly seen in the next slide. Here, when we compare ionophores on their own versus ionophores and SETI, we can see how for 160, the activity was more than double. For 158 and 159 was almost double. And then for 157, there was little or no increase in activity. Thus, we can see how these DIF-OPBU and ionophores restore anion transport activity, and their action was synergistic with NMD inhibition and CFTR modulation. Then we wanted to see how this combination of DIF-OPBU and ionophores and NMD inhibition and CFTR modulation has affected the cell viability. Then we did some XTT assays, and here we can see how ionophores on their own with compounds 158 and 159 show some degree of cytotoxicity, but 157 and 160 they were non-cytotoxic at all. When we compare with an ionophores combined with SETI, all of the compounds show some degree of cytotoxicity. Therefore, this combination of di-FOPBU and NMD inhibition and CFTR modulation increase cytotoxicity. Just in summary, we have done some small structural changes in the compounds 158 and 159 that vary disfluorination levels and rigidity, increase anion transport activity. Di-FOPBU and ionophores were more active when they used in W1282X CFTR HB-YFP cells than in FRT-YFP cells. And when they were combined with NMD inhibition and CFTR modulation, there was a synergistic effect. And the cytotoxicity studies showed that di-FOPBU and ionophores was increased when they were combined with NMD inhibition and CFTR modulation. Therefore, our future work will focus on developing inhaled-controlled delivery of di-FOPBU and ionophores to achieve good anion transport activity, lower doses, and less cytotoxicity. And that's to say thank you very much, everyone.
I assume that the chloride, the anion movement in the presence of the ionophore is electrogenic. Is it carrying a current?
What do you mean by
current? Is it electrically neutral or is it charged because of the chloride? I don't understand the chemistry well enough to know whether when the chloride and that molecule move from one side to the other side, is it changing the voltage? Is it carrying a current?
I mean, it's following the chloride gradient, but it's not showing the current itself, because when we have done using chamber experiments, there wasn't a disruption on the current, but when we can clearly see that exchange of chloride by using the fluorescent assays, then that helps.
Okay.
Thank you.
A very interesting data set. Do you have any data on bicarbonate?
No, we haven't done that acids yet.
It's just an interesting opportunity, even just fundamentally to probe role of chloride and bicarbonate in restoring whatever other properties, look at pH or viscosity, etc. If they're selective for chloride and they don't transport bicarbonate, it would be very interesting to run the experiment to try to help understand how chloride and or bicarbonate contribute to those other downstream outcomes. So very interesting.
Yeah, we probably will do that in the future. Thank you. Thank you very much. If any other questions, please visit the poster 260. And yeah, I wanted to say thank you all for everything.
Okay, so this concludes our session. We want to thank you so much again for taking time to visit. Thanks for all the great questions and discussion. I think we end where we began, which is there is an urgent need to address this issue. And I think it's going to require a lot of different thinking, a lot of shots on goal, and a lot of persistence. Okay, it's a very challenging problem. So let's keep going and let's keep trying lots of different ways to approach and ultimately find a way to treat everyone with CF who are waiting for those treatments. Okay, thank you so much again for the time.